Alrighty, well, we're 3 minutes past the hour, so we can make a start, I think. So, good afternoon, everyone, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. With this series, we hope to shake up the usual format of lectures by having a mixed format with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Now, typically these sessions will run for about 50 minutes in total, and we'll run the series throughout the year and generally on the last Thursday of each month, unless there's some overlap with holidays like we do today. If you are interested in presenting a new method or software you have been developing or implementing, please feel free to reach out. And before moving on, just a few quick introductions. So my name is Lewis Jones, and with me today I have Erin Dillon, our new Paleoverse member Harriet Drage, and William Gertie. And the four of us are on the development team of Paleoverse, an organization aiming to bring the paleobiology community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. And if you want to know more about Paleoverse, please check out our GitHub organization and our website. And I think those links will be dropping into the chat now. But that's all from me for now. Harriet will now give you some helpful tips and instructions for this webinar, followed by Erin, who will introduce this month's speaker. So thank you very much, and please enjoy the lecture. Over to you, Harriet.
Hi, everyone. So as Lewis mentioned, the format of this seminar is going to consist of a talk by our speaker. Then we're going to follow this with about 10 to 15 minutes at the end dedicated to a Q&A and a discussion. My fellow hosts and I will help you field your questions and moderate all of this discussion. Please note first off that the seminar will be recorded and be made available on the Paleoverse YouTube channel, but we'll stop recording before the Q&A takes place in case you prefer not to be recorded when discussing. Before we get started, I'd like to point out a couple of useful controls at the bottom of the screen. First off, we have a Q&A button, so you can type in here any questions you have for our speaker. And then during the Q&A session at the end, we'll either read these out or invite you to turn on your microphone if you'd like to ask your question directly. Alternatively, there's also a raise your hand button, which you can use in the Q&A period, and then we'll call on you to turn on your microphone and ask your question. There's also a chat button. At the minute, you can only chat with the hosts in case of technical difficulties. We'll allow you to chat with the other attendees as well during the Q&A period. And lastly, there's a closed captions button, so you can turn that on and view the transcript at any time. Finally, I'd like to highlight a couple of ground rules for the webinar. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can learn from each other. With this in mind, we ask you that you please be respectful and kind when engaging with the speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behaviour will be grounds for removal from the webinar platform. For more information, please refer to the Paleoverse Code of Conduct, which can be found on our website, and there should be a link going in the chat for that as well. And with that, I'll pass over to Erin to introduce today's speaker.
Awesome. So thanks so much, Lewis and Harriet, for getting us all up to speed on how the lecture series will work. Today, I have the great honor of introducing our speaker, Dr. Roxanne Banker. Roxanne is a new assistant professor at Providence College in Rhode Island. She completed her PhD in the Department of Earth and Planetary Sciences at UC Davis in 2019 and went on to do postdocs at the California Academy of Sciences and at the University of Nevada in Las Vegas. Roxanne's work synthesizes modern and geohistorical data and applies mathematical models to answer fundamental questions in evolutionary ecology and inform practical conservation strategies. In particular, she is interested in studying ecological processes that influence the resilience, stability, and persistence of marine species and systems across various temporal and spatial scales, and in response to climate change. One really neat example is her research reconstructing trophic networks of paleo communities using fossil data and testing how they respond to disturbances. So today we're really excited to hear from Roxanne with a talk titled Quantitative Approaches to Understanding Marine Ecosystems, Insights from Stellar's Sea Cow and Ancient Food Webs. Please join me in welcoming Dr. Banker to our virtual stage. Take it away.
Thank you so much for that wonderful introduction, Erin. And yeah, it's great to be here. Thank you, everyone who came today. Slide, there we go. Yeah, so as Erin introduced my talk title, I actually realized I wouldn't have enough time to sort of get into the methods of both stories. So unfortunately, if you're here for the sea cow, I'd be happy to talk about it later. But today I'm actually just going to be talking about some of the ancient food web reconstructions that I've done. All right, so we're all, I'm sure, very familiar with the idea that modern ecosystems are under threat. I'm in Rhode Island now, but I just spent the last 10 years doing research in California. And so some of the sort of disturbances that marine communities have experienced on the California coast over the last 10 years include thermal shock. So there have been like really big mussel die-offs due to higher temperatures, harmful algal blooms. We've also dealt with an outbreak of sea star wasting disease, which there's a picture on the bottom left. Oops. And if you're not familiar, that's a pathogen that sea stars acquire that really just causes them to disintegrate. And the removal of this keystone predator on the West Coast has had a lot of far-reaching consequences for those environments. A more sort of human example on the right is a picture of the San Francisco, what is that? That is the Golden Gate, I think, or the Bay Bridge. But anyways, there was a day in 2020 when we were experiencing forest fires and the ash, what we're looking at here is there is actually ash in the upper atmosphere, so the air on the ground was breathable, but for a full day, we were just living in this red light. So the consequences of climate change are real, and they're here, and we're all experiencing them. And what I'm really interested in my research is this idea of a shifting baseline. And so as humans, human generations are progressively more used to increasingly degraded environments. So here's a nice example. On the left side is a picture of a fishing charter from 1957. And you can see that the largest fish they caught were as large as this family's child, right? These are huge fish. The same charter went out in 2007, pictured on the right, and you can see that their largest catch from that trip was a small shark. And then there's a human man's hand circled just for scale. So again, a shifting baseline is this idea that we're progressively more used to these degrading environments, but we don't necessarily realize it because we're only used to what we've experienced ourselves as individuals. And so one other way we might experience this is through changing community composition over time. So in this example, on the bottom, say oceans around the 1800s, we might have seen a great amount of faunal diversity in the sea and coastal ecosystems. But then as we move to 1950 and then 2019, the community composition has changed a lot. And what we go out to see is going to be a lot different than what our ancestors saw when they went out to the same environments. And so what I'm really interested in my research is introducing historical data, paleontological data to try to better understand ecosystem dynamics and specifically what are the role of biotic interactions in ecosystem dynamics. And so on the right is a nice figure from Tyler and Schneider 2018 that shows sort of like the different levels of historical data that you can bring into your research. So we might consider more human-influenced timescales in these two first circles in the yellow. The past 500 years might include environmental history, historical documents, records, personal journals, things like that. And that actually is a lot of the types of data that we used to try to reconstruct how Steller sea cow influenced kelp forest ecosystems. But I won't talk about that a lot today. And ultimately, what I'm really interested in is, like Erin said, how do ecosystems respond to perturbation or disturbance? And how can we identify properties of ecosystems that might actually promote stability in the face of disturbance and long-term persistence in the face of, say, climate change? And one of the other tools that I use in my research that I'll be talking about more today are food webs. And so a lot of our current ecological understanding of systems really comes from community composition. So when we go out into the field, who's out there? What organisms are we looking at? And that's really necessary information, but the number of species doesn't necessarily equate to the number of behaviors or the number of ecological functions in an ecosystem. And it turns out that the diversity of functions in an ecosystem and how they interact with one another is really important for the stability and long-term persistence of systems, because it affects energy flow and a lot of other emergent properties of systems. And so here's just one example on the bottom of the slide. So a really simplified food web diagram might be represented where nodes or circles represent species or other entities, and interactions are usually drawn as arrows going from the prey to the predator, so in the direction of energy flow. And then here's a more complex diagram that other folks who, especially those from the West Coast of the United States, might recognize. So this is a kelp forest ecosystem. So some of those nodes might represent kelps, some of their predators, going all the way up to what we think of as the top predators in systems. So in this case, sea otters, sharks, and other large fish. And so today I'm really going to be digging into this project where my group, so many of these folks here, we reconstructed Mesozoic food webs to better understand what community level changes were happening when we see these other large scale changes in the environment. And so I just want to call out Dr. Peter Rupnerine, who works at the California Academy of Sciences, Dr. Ashley Deneen, Dr. Carrie Tyler at UNLV, at the University of California Museum of Paleontology. There we go. Dr. Carrie Tyler at the University of Nevada, Las Vegas, Carrie's PhD student, Madeline S., and then a number of other master's students and undergraduates who helped in this project also. And so what I'm really interested in this project is bringing in some deep time data. So the fossil record offers some really incredible opportunities to better understand how these ecological processes that we're interested in the modern and in the context of climate change, how do we study these processes over evolutionary time? And we can do that by going back into the fossil record. So one of the really big questions that I had during this week, over the course of this project, was, does changing predation intensity and biodiversity correspond with changes in community structure? And so for this, again, we worked to reconstruct food web networks of ancient ecosystems from the Mesozoic. And again, we're bringing in some nice deep time data, fossil evidence that I'll talk more about how we collected that. And so for anyone who's not really familiar, the Mesozoic marine revolution was this really interesting period of geologic time. So this figure shows time, again, from about the Cambrian 540 million years ago to the present. And on the y-axis are the number of marine families. And again, the Mesozoic marine revolution was this period of time where we see not only a really significant increase in the number of marine families, but we also see a huge increase in the number of different ecologies and functions that organisms are performing in the environment. And so on the left is a figure showing, again, geologic time. The Mesozoic is highlighted in this dashed square. But this is really when we start to see the radiation of shell-breaking cephalopods. We see the expansion of marine reptiles, the radiation of shell-crushing crustaceans, shell-breaking fish, sharks and rays, 4th century celeroids. So like all of the big predators that are really important in modern ecosystems were really coming online during this time. And we think that a lot of this is due to an increase in primarily planktonic primary production during this time. And so here's another figure on the right, and on the x-axis is, again, geologic time from 252 to 0 million years ago. The Mesozoic is, again, squared off in that purple dashed line. And the gray line shows a strontium isotope ratio, which is really a proxy for terrestrial runoff, so nutrient input into the ocean. And then these different lines show the diversity of different planktonic primary producers. And you can see that particularly the coccolithophores and the dinoflagellates were really expanding in diversity starting in the Triassic all the way through to the Cretaceous, and diatoms started coming online in really big abundance in the Cretaceous, or in richness. Right. And so we really think that the Mesozoic marine revolution was supported by this increase in the primary producer base during this time. And so for this work, we're actually taking fossil data from marine communities of the Western Tethys. And specifically, we were able to collect data from 4 different ages throughout the Mesozoic: the Anisian, the Carnian, the Bathonian, and the Aptian. And this was essentially because these are all of the time periods that we could really find sufficient data for to create these food web networks. So now I'm going to break into a little bit more about the workflow, particularly the data preparation sort of side of the workflow, in how we build these food web networks, these food web models. And then in a few minutes, I'll dive back into the results of some of this work. And so here's a really nice, here, I'll skip here because the citation's on this slide. And so this is a really nice schematic that was created by Younggang Wang, Peter Rupenrein, and their other collaborator. This paper is still currently under revision in Nature. And it just shows a really nice sort of schematic diagram of the different steps that we use to reconstruct food web networks from ancient ecosystems. And the first step is this data preparation step. And so what we really want, or what's really important here, is considering your question, the geographic and temporal scope, because that'll really affect what kinds of questions you can answer with these food web networks. And so really the goal is to get a taxonomic list of all of the organisms that were in your location of interest over some defined time bend. And you really want this list to be as comprehensive as possible. So once we have that taxonomic list, we go through the process of ecologically parameterizing all these species. So for each separate species in this list, we assign these species to ecological guilds based on ecological properties that they share with other organisms. So this includes locomotion, are they nectonic? Are they crawling? Are they moving in some other way? Are they sessile? Habitat, are they benthic? Are they pelagic? Common predators and prey, so are they eating the same things? Are they being eaten by the same things? And then also body size is a really important one, because of course, body size will constrain how big your mouth is, and then who you can eat and who can eat you in turn. And so, again, this first step is taking them to this guild framework. And then we sort of synthesize these guilds, and again, we do a lot of research, a lot of literature review to figure out, okay, what other taxa were they eating? And so we can start to sort all of our species into these defined guilds and based on common predators and prey. So here in this example, this row, omnivorous benthic microzooplankton. We might have a few different species included in there, but they all have or would prey on members from guilds 1, 2, 3, and 5. Using that information, we build a meta network or a guild level food web for which guilds were eating who in this ecosystem. And so here's an example of what a guild level network would look like for a modern system. So sorry about that. Some of the producers would be here at the bottom, moving up to microzooplankton, up to larger predators like cephalopods and fish. And so again, we're not identifying predator and prey species, only predator and prey guilds. And those interactions are then assigned between guilds, which gives us a meta network. And so we can end up with this final document that we end up using in subsequent data analysis. So this would look like our list of guilds, and this is actually from the Mesozoic data set. A higher taxon, this is really helpful just to keep track of sort of who's who in this data set. The number of species, so richness of the guild, and then again, their different prey resources. So what other guilds are they feeding on? And here's just another example of what this looks like. Again, this is our actual data from the Anisian, Carnian, Bathonian, and Aptian. So again, their prey resources are listed in this data table. So now we have our condensed form of the guild assignments, and this can be used in R itself to actually build visualizations of the meta network and to build the interaction matrix, which is an input file for the larger model. And I'll talk about that in a moment. Right, and the guild list specifically is formatted to have 5 columns. Again, this is how it's read into our subsequent model, where there are 5 columns listed below, and the number of rows is equal to the number of guilds. And so column 1, as I showed on the previous slide, is guild number. Column 2 is guild name. Column 3 would be the dominant higher taxon, which is really just helpful for, again, sort of keeping track of who would be in that guild. Guild richness, and then the list of resources. So who are their prey guilds? And then finally, we use that information to create an interaction matrix. So this is where I believe predators are on the y-axis. Yeah. A presence-absence matrix with k rows and k columns, where k is just the number of guilds. And rows are consumer guilds, and columns are the resource guilds. So if any given cell has 0, that indicates that consumer in that row isn't feeding on that prey item, whereas a 1 indicates that guild is having a predator-prey resource interaction. And so here's an example of what that would look like. Oh, sorry, my mouse is too sensitive. And so here, Guild 6 does feed on Guild 3, but it's not feeding on Guild 4, for example. All right, so now we've gone through the process, we've assembled our taxon list, we've constructed the guilds, and we've built a guild meta network, which also helps us get to our interaction matrix. And in step 3, we actually create a guild parameters list. So this is, again, a 4-column file where there are k rows, k number of guilds. And this is one of the data sets, again, that's fed into our model in the Julia programming language for modeling and analyses. So again, the column 1, guild name, column 2 is usually the higher taxon. That's, again, a helpful placeholder to keep track. Guild 3 is species richness of the guild, and column 4 is a general trophic position term like producer, consumer, depending on how you code it, which again will help you build color-coded sort of visualizations in the meta network. And so overall, what we've gone through so far are these different data preparation steps leading up to the model with the food web reconstruction model. And so again, just in summary, this includes data preparation, so the taxonomic list, aggregating all of those taxa into guilds, collapsing the guild list, producing the adjacency matrix, and creating those guild parameters. And these last 2 are what gets fed into our model in Julia. And here's a visual representation of what that looks like. So again, our taxonomic list, we go through and parameterize each species. We decide who their predators or prey are, and this helps us to create the input files for the model. And I just want to pause here too to highlight some previous work done by Peter Rupnerine and Rachel Hertog, and then in a subsequent paper, they collected the data for this work. In a subsequent paper, Peter and Ashley actually reconstructed the food web networks from this Caribbean data set. And I just want to highlight that this data set consists of 728 species, 249 guilds, and over 4,000 interactions. Here's a visual representation of that network. And what's really cool is what their comparison showed is they actually analyzed the modern Caribbean reef web, and they compared it to a predicted fossil web. So they just removed all of these soft body taxa that wouldn't make it into fossil records, specifically taxa that don't have a record in the PBDB. And despite the loss of soft-bodied organisms, overall gill diversity, community structure, and other parameters, food web network parameters like modularity remain intact. So that's just to highlight this model system. If you go through the steps of creating your taxonomic list and guild list very carefully, these models really do reconstruct the diversity and food web structure of even a really complex modern ecosystem. And so the way that we actually go from guild level meta network to species level networks, because we're all paleontologists and geologists here, we're very limited, unlike ecologists. We can't go back in time and observe who is eating who in an ecosystem. And so we go through those steps of creating the guild level network, which will help us constrain and estimate species level network topologies. And so what the big model does, and I'm not going to go into a lot of detail about the model itself, but I'm happy to talk about it more. But essentially what the model does is it takes our guild level structure, and we take each of the taxa from each of the guilds, and in each sort of estimate of what the species level network would look like, links are randomly drawn between prey and predator species, but always consistent with that guild level meta network topology. So in this example, you're never going to have the pink star having a resource link to the orange square. These links are always going to be consistent with that guild level network that you constructed. And so the reason we do this is, again, you can't observe. So we're using the guild level network to estimate usually about 1,000 species level networks for each community to overcome that uncertainty statistically. And so then what we can do is we can study each one of these species level networks and look at different food web network parameters. And two of the parameters that I'll be focusing on today are guild maximum chain length. So what's the maximum chain that a given species will find itself at the end of? So this square is at the end of a 1-2 step chain. Maximum chain length in all of these is about 2 steps, I think. Whereas network trophic position, I'll explain here on the next slide. Network trophic position is an interesting metric. And here's the mathematical equation, but qualitatively, I like to think about it as the average shortest distance of a predator's prey species to a primary producer. So here's a figure that I adapted from one of our recent papers. And so they're both both taxon 1 and 2 are at the end of food chains that are 1, 2, 3, 4 steps long, 1, 2, 3, 4 steps long. So they have the same maximum chain length. But taxon 1 is only feeding on this other taxon that has a relatively high trophic position. In contrast, taxon 2 is feeding on other taxa lower in the food chain. So taxon 2 has a lower network trophic position than taxon 1. And we can measure the distributions of parameters like maximum chain length and network trophic position to try to get an idea of how ecology was changing in that community at that time. And I'll show you some of the data from the Mesozoic data set here now. So here is a Bayesian linear regression of guild maximum chain length on the x-axis and guild mean network trophic position on the y-axis. And what's really interesting, I think, is the Bathonian has a distinctly higher slope in this graphical space, Whereas thinking about the Mesozoic marine revolution or the Mesozoic, you really see like an accumulation of more energetically expensive lifestyles. There is a bit of a direction to it. So then the question becomes, why does the oldest community right after the and Permian extinction, why does that have the most similar structure in the space to the Aptian, which is our most modern community in the data set? And so what we think is that this is really to do with the balance of long versus short food chains in the ecosystems that we can see from these reconstructed food web networks. So again, just to go back to that figure, taxon 1 might have a relatively higher network trophic position because it's feeding high in the food chain, whereas taxon 2 has a relatively lower one because it's feeding closer to that primary producer base. But they both still have a maximum chain length of 4. And so what I think is happening is during the Bethonian, what we see is for any given maximum chain length, though those organisms were feeding higher in the food chain. And I think that this is to do with the types of predators that were really dominant in the Bethonian versus the Aptian. So in the Bethonian, this is really when we see, especially in our data set, a sort of peak in marine reptile diversity. These were the main predators in the Bethonian. And it makes sense that those really big predators would be feeding on other things that are relatively large and likely specialized also. By contrast, in the Aptian, we start to see sharks becoming a more dominant predator in terms of their abundance in our data set. And in modern ecosystems, sharks are really important mesopredators or trophic omnivores. They do a lot of this feeding behavior where, yeah, we think of them as top predators. They're very ferocious, but they are also feeding on a lot of other organisms in the food chain. And so if we put this into context again of what was happening environmentally at this time, we hypothesize that this pattern in the Bethonian and the reason that it sort of went back to an Aptian sort of slope in those previous graphs is because of the enhanced primary production in the Jurassic as compared to the Triassic. We hypothesize that sort of these new primary production resources coming online enable the expansion of these really big Mesozoic marine reptiles, but this perhaps led to unstable sort of top-heavy food chains. So then trophic omnivory started to re-expand into the Aptian, which is why we see more of those shark mesopredators in that system. And so if we go back again and compare the Mesozoic data to our modern Jamaican coral reef, we can plot that onto the same line, and it looks a lot like the Bethonian. And I thought that was really interesting. So I think and what's really notable is that there's been about a 71% decline in shark abundance since the mid-Holocene, and I don't know what happened, but I was citing Erin's paper here and I don't know where her citation went. So Erin Dillon, this result. I got that from one of her papers, I know. And so given this recent shark decline, my question is, would the structure of a pre-human Jamaican reef community look more like the Anisian and Aptian, where we're starting to see those sharks in the Mesozoic? And is it looking more like the Bethonian because we've removed a lot of those mesopredators and thus sort of shifted the balance of long versus short food chains? And so I just want to wrap up now. I think there's a lot of really interesting insights that can come out of looking at these ecosystems from a community food web level perspective. Natural systems are really complex and they respond to change in nonlinear ways. And so I think food webs are a really powerful tool for starting to dig into some of those questions. And it also just highlights a need to better understand the relationship among global change, community functional composition, and other emergent community properties like long-term stability and persistence. So with that, I think I'm on time and I'd be happy to take any questions.